Friends, welcome to Biology Made Easy. Today in this video, we are going to discuss the complex permanent tissues. Previously, in the first video, I have told you that the complex permanent tissues are two types. One is xylem, the other is phloem. So today, first we'll discuss about the xylem. Now, xylem is otherwise also known as wood. The secondary xylem is known as wood, and it is also known as hadrome. Hadrome by Haberland. As you know, that the main function of the xylem is conduction of water and minerals from the root to all the upper parts of the plant body. But what are the different constituents? Xylem is a complex tissue. Here there are four types of cells and all are different from each other. That is why it is a complex tissue. Whereas in parenchyma, collenchyma and sclerenchyma, the cells were of one type. That is why they are simple. But here we will get different types of cells. And the four different types of cells that are present in the xylem are number one is tracheids, number two trachea, or we can call them as vessels. Then number three is xylem fibers, and fourth is xylem parenchyma. These are the four different types of cells that are present in the case of xylem. Out of these four, the first three are dead. These are the dead tissues, while this one is only living. Now we have to discuss the structure of each one. Now let us discuss first the tracheids. Tracheids they are present mostly in case of pteridophytes and gymnosperms. This is the major constituent of xylem in case of pteridophyte and gymnosperm, while only 5% is present in case of angiosperms. Now these are long, elongated and tubular cells. Long, elongated and tubular cells. Next, the ends of cells are tapering. Next, the end walls are imperforate. Imperforated means there is no connection between different types of tracheids, and we'll discuss in the diagram also. Then the cells are very thick, and the cell walls are highly lignified. They are highly lignified, and because of this high lignification, because of the high thick and high lignification, the lumens are narrow. Now if we draw the diagram, the cells will be long, narrow and elongated. So this is one tracheidal cell. Suppose another will be there, it will be like this. Then another is there, it may be like this. These are the cells, and there is no direct perforation between them. And the cell walls are very thick and lignified. They are very thick and lignified. And the inner space is known as lumen, because these cells are dead and there is no protoplasm. cells are dead. So there is no protoplasm. That is why the space is known as lumen. Now, between the thickness of this cell wall is again of different types. The wall thickenings are of different types. And what are these types? It may be annular, or it may be spiral, or it may be scalariform, or it may be reticulate, or it may be pitted. These are the different types of thickenings. Now, suppose this is one tracheid, and the thickenings are present like this. Then this is known as annular. Then, if the thickening is present just like that of the spirogyra, this is known as spiral shaped. If it is present like a ladder, the thickening, then it is known as scalariform. And if it is present in a net-like manner, then this is known as reticulate. And if there are pits like this, pits are there, this is known as pitted. Out of these different types of thickenings, the annular spiral, they are quite primitive type of thickening, while pitted is the most advanced type of thickening. Now the pits are again of two types. There may be simple pits or bordered pits. But in the tracheids of gymnosperms, large number of bordered pits are there. So let us draw the diagram. Suppose this is one cell, and this is the inner one. Then this side, this is another tracheidal cell. Now you see, here the secondary wall is not deposited on either side. This is known as a simple pit. This is simple pit. And the simple pits are usually present in the simple tissue like parenchyma, collenchyma, sclerenchyma. But in case of tracheids, usually bordered pits are found. Bordered pits are found. So the bordered pits are like this. Now here we'll find some bulging. Here also on this side we'll get bulging. And in between we'll get some, here we'll get some thickening like this. Thickening, this is made up of suberin, and this is known as torus. This is known as torus. It is made up of suberin. And this bulging sheet, this is known as bordered pit. And in the tracheids of gymnosperm, these bordered pits are there. Now what is the function of this torus? It is made up of suberin. Sometimes there may be more flow of water in this one of the tracheid than the other one. So in that case there will be heavy pressure on this middle lamella, and there is every possibility that it may break down. That is why the torus, which is made of suberin, it controls it so that there will not be heavy pressure and the middle lamella and the primary walls will not break. So it provides one type of protection while water flows from one to another. So these are the different types of the border pits, and these border pits are present like this. So this is one, and this is another. Here suppose one pit is there. So this border pit, this border pit will be found here. Here also one border pit will be there. That means here one border pit is there. Here one border pit, several border pits are there. And again you mark. These are not they are not imperforated. They are connected only by means of these pits. So when water moves, water will move from, it will move to this one. From this it will move to this one through this pit only. And from again to this it will move through pit. So it will move in a zigzag manner. So this is the structure of the tracheids, which is predominant in the xylem of pteridophytes and gymnosperms, and very less quantity, that is up to 5 to 10% present only in case of angiosperms. So this is the structure of the tracheids. They are usually this narrow, elongated and with tapering ends. But now you come to the vessels.